That's good. Do you see my presentation? Yes?
Yes.
Yes. Okay. I want to present my ABC transporter Accosited Chemoresistance of Tumor cells. All living cells able to develop resistance to different undesirable for them factors, including cancer cells resistance to anti-cancer drugs. And the main function mechanisms of this resistance of cancer cells is ATP-dependent transmembrane proteins of ABC family activity, P glycoprotein and breast cancer resistance protein. This protein's activity in healthy cells leads to xanabiotics and metabolites efflux, and in cancer cells to protection cells from different kind of anti-cancer drugs. And ABC transporter inhibitors are used for ABC transport, and the action of mechanism of action of these structures is binding with inner part of protein, transmembrane domain, but unfortunately it hasn't been able to show efficient clinical results, and we think that its approach has exhausted its possibilities, and it should be changed. And we draw our attention on the other structure of protein, which hasn't studied in the nature of ABC transporters inhibition, nucleotide binding domain, which in the same time is responsible for ATP dependent ATP binding, its hydrolysis and transporters activation. So we assumed that ATP replacement by structures which are structurally similar to ATP would help inhibit transporters by NPT blocking. So to task this hypothesis, we consider AMP and compounds with a ribose residue and nitrogenous base. So the main aim of this work was to validate NPT as a new target of ABC transporters inhibition by studying compounds interaction with ATP with ABC transporters, and by evaluating compounds biological activity. And the first for testing for ATP mimetics interaction with NPT evaluation was realized by computer modeling with via semi-rigid docking with NPT of both proteins. And it was revealed that interaction with TMD of PGP and BCRP has low scores, and interaction with NPT, on the other hand, high scores. And the best results showed the closest analogies of ATP, which stacked, reproduce stacking of ATP in NPT of PGP and of BCRP. By realizing of luminescent analysis using recombinant PGP, was revealed that the presence of compounds leads to proteins ATP phase activity inhibition. It means that protein system consumption of ATP decrease. And the biological activity of compounds were carried out by using human colon adenocarcinoma cells line, which characterized resistance of high concentration of taxol and or expression of proteins genes. And it was revealed that ATP mimetics adding made it possible to reduce transporters activity and increase accumulation of nuclear dioxin cells, increase sensitivity cells to taxol, leading to cells die. So The obtained results prove that nucleotide binding domain is the new potential target to overcome cancer drugs resistance because ATP selective binding to NBD leads to transporters ATPase and transport activity inhibition and chemoresistance cells drug overcoming. So
Okay. Now there is some questions on the chat. One of them is what docking program was used from Professor Sulimov?
yes, we used a set of Schrödinger program.
Okay. Roman Yefremov asked how the membrane environment was modulated docking to TMD.
Sorry, I participated during aim and task definitions, and computer modeling directly was realized by my colleagues, and I may clarify this moment and task contact with you later.
Another question from Professor Sulimov. Did you use PDB structure to prepare a protein model for the docking?
Sorry, can you repeat the question, please?
Did you use PDB structure to prepare a protein model for docking?
Yes, we used.
Professor Paroikov, do you think the difference between the 7 and 8 value scoring function is significant? Explain why.
Sorry, with this kind of questions, I should to address my colleagues. Thank you.
Okay.